Now, we will discuss another very interesting and very precise technique which is used in the molecular biology, and that is microchip electrophoresis. As the name implies, microchip electrophoresis, it allows very high speed analysis at very low sample sizes. Now, mind you, when we are doing analytical chemistry or analytical procedure, there are two things we have to consider. One is the speed, and second is the size of the sample. Now, in microchip, as the name implies, the very high speed analysis can be carried out with very small concentration of sample. Now, we have seen, or we have previously discussed, the capillary electrophoresis and the conventional gel electrophoresis methods which are there. Now, how we compare them? Now, microchip analysis, that can often be completed in 10 seconds, very fast, whereas capillary electrophoresis can take 20 minutes, and the conventional gel electrophoresis, that takes at least 2 hours to complete the analysis. So you can find out, or you can see that what are the advantages of microchip electrophoresis. Now, in this case, the applied voltage required is only few volts, which eliminates the need for the high voltage which is used by capillary electrophoresis, or CE. So we see in the diagram that the capillary electrophoresis, it requires high voltage so that the molecule transfer. But in the capillary electrophoresis, the current needed is extremely low to few voltages. And the manufacturing process that produces microchip is called microfabrication. Very simple composition, that how we can, as a matter of fact, make a microchip. Now, the process of microfabrication, that involves etching of the capillaries very precisely and reproducible manner, just like capillaries, channels on the surface of sheets of quartz glass or plastic. So we take the quartz glass or the plastic and etch on it very precisely the capillaries which are there. A second sheet on that plastic sheet is then fused on the first sheet, and it is turning the etched channels into closed microfluidic channels. So you see, we have got the capillaries, small capillaries etched on the sheet, and then we put another sheet on that so that it makes a small micro channel there. Now, the end of each channel, it connects to the reservoir through which fluids are introduced or removed. Just like that, the end of these is dipped into the reservoir containing some fluid, and then it introduces that fluid into it. Now, basically, the microchip, it provides an electrophoretic system which is similar to capillary electrolysis, but with more flexibility. You see, the capillary electrophoresis is very cumbersome, large equipment. You have to introduce a sample. But this is the chips which you have made, microchip. And mind you, on one sheet, you can put lot of those capillaries, and you can attach them to the other end. Now, what are the current developments in this technology? That is, the development are that it is based on integrating function other than just separation into chip. That there are a lot of other integrating or the combining function which can be achieved, or which are the advantages of this technology. And for example, sample extraction. We can have pre-concentration of samples prior to separation. That is, the sample which is introduced can be concentrated prior to the separation. We can also amplify that through PCR amplification of DNA sample. That is, the DNA sample, first it can be subjected to the polymerase chain reaction so that the DNA concentration increases, and then we can run this. Now, an interface has also been developed for microchip electro-phoresis, mass electrophoresis, between mass spectrophotometry, MCE, mass spectrometry, and mass spectrometer. That is the interface. That is, they have been joined together, these two techniques. And when we need, and where this is used, when we need that drugs have to be separated by mass spectrometry and then identify by mass spectrometer. So the interface with the mass spectrometry and the mass spectrometer of this microchip electrophoresis can be devised, and that can be used where we have to identify the drugs, different drugs which are there or other. So you see that these techniques, they are used frequently for analyzing the metal, for analyzing the protein, for analyzing the DNA, and so forth.